Welcome to Talk Universe. This is our show for Wednesday, October 19th, 2016. And tonight we have the subject of genetically modified organisms on the table. Originally, we had an interview scheduled with a NASA engineer who creates genetically modified organisms for specific purposes, and scheduling difficulties arose. So we're going to put that interview and that show off for another time. And we're going to look at just exactly what a GMO is and what the difference between genetic modification and selective breeding might be. Now, that's a very important topic, and a lot of people don't understand the difference. So we're going to have a look at the mechanisms behind selective breeding and making new plants from other plants or new animals from other animals, and what the genetic modification controversy is all about. Now, suppose you're walking in the woods and you're looking at the plants and animals all around you, and you begin to wonder as you're walking around out in the wild, just exactly where do you find food? Now, there are some things that we can see, such as blueberry bushes or wild turnips, or some of the things that we spot here and there if we have an eye for it and we are familiar with the plants themselves. There are things we spot that we know we can eat. Obviously, anything that grows on an apple tree is pretty much going to be an edible fruit. But you wonder, where did all that we have come from? As you're walking through the fields, you never see a wild string bean or a wild tomato plant. You rarely, if ever, see anything that looks like a carrot. And you have to wonder, where did all the food that we have actually originate? We know of the difficulties of raising vegetables if we grow a vegetable garden, and we know that it isn't something that simply happens on its own. That in fact, the plants that we eat often are impossible to grow in nature without a great deal of help. And that is why we do gardening the way we do. So the question is, where did these plants come from if they don't grow freely in nature? Now, to understand how all of our food crops came to be, we have to understand a little something about genetics. And believe it or not, the food crops that we have are not, as a rule, genetically modified, even though you don't find their cousins easily in the wild. There are plants in the wild that many people years and years ago knew were edible, but they are nothing like the plants we have today. And so as you're wandering through the woods, you don't see fields of wild squash and fields of wild melons. The obvious answer is we've done something, and that's true. Genetic modification and selective breeding are two of the tools that can be used to create new plants and animals. And we're going to look at exactly how that occurs, and understanding the genetics behind it is an extremely important part of this. You see, when we look at genetics, we see that there are two obvious things, and one is the genotype, the actual genes themselves that an organism has, and the other is the phenotype, and this is what the organism looks like, its structure, its appearance, its function. So genotype and phenotype are tied together, and these are terms you probably heard in high school or college. But it's not an obvious relationship in some ways, and people had to explore for a number of years to understand the connection between the two. We know that there are families of people who have red hair, and it seems to run in the family. And this is the phenotype. You're seeing the red hair or the freckles that go with it. But if you wonder about the mechanism behind it, you obviously have to look at the genes themselves. Having a gene for something doesn't mean that you will have that trait or characteristic. Many people have genes for things that are not expressed at all. And so the question arises, what makes a gene express itself? How do you know what other genes are hidden in there? And what does it all come down to? So let's go back to the beginning and have a look at how food crops came to be, and we'll look at the mechanisms behind getting those foods to be what they are today and how the understanding came about. In the distant past, people would select the plants that they knew were edible, and when agriculture arose, they tended to begin cultivating them because then they would have a supply right on hand. One of the things that began to happen very early on is some of the plants, of course, are not identical to the others, and you will often have plants grow in a variable manner. Some are larger, some are smaller, some are tastier, and what people would do is select out the best ones, the ones that grew a little easier, had a larger yield, tasted better. They would select those. They would save the seeds from them, and those are the ones that they would plant. And as a result, those traits were the ones that were carried on to the next generation. And so each time that they would raise their plants, they would save the best ones, the seeds from the best plants, and those plants would be planted. And it only takes a few generations for a plant or an animal to become domesticated. Now, what is domesticated? In essence, domesticated is a little more friendly for our use, whether it's taking wolves and breeding them into different breeds of dog, or if it's taking a wild plant and accentuating its traits so that it produces a better yield for us. And typically 7 generations is the figure that is quoted, because in that period of time, a trait has an opportunity to be reinforced in a way that we like, and we have the ability to weed out some of the undesirable traits along the way. But an individual doing this is one thing, and a farming community doing it is another one entirely. You see, when an individual does breeding of this sort, there is a small sample size, a small number of organisms that you're working with, and one person or one family who is concentrated on doing this. But when you have a community doing it, you have many, many more plants and a higher chance that some variable factor is going to show up, like a larger grain or a sweeter apple. And this means that the resources of a whole community can be concentrated on producing what is best. Not only that, when you have more farmers doing this, you have a better opportunity for their different planting and harvesting techniques to work properly. But there's another factor that comes into play, and it's called religion. When a certain rule is discovered to work a certain way, you often cannot force individuals to follow that rule. But if you have the rule of law or the rule of religion to go by, it becomes much easier. And one of the earliest tools that helped us shape agriculture and the crops we have was the thought that the best of the crop was picked and placed on the altar for a blessing by the gods. Now, what was actually happening was the best of the crop was being selected out and its seed was being saved. And it's an interesting link between religion and agriculture, because early on the church acted as the bank for storing your grain or storing your seeds after a harvest. And this concept meant that often, whoever owned a specific lot of seed or a specific type of plant would end up with the genes mixed with the other farmers' plants as well. And so traits would become amplified in a small gene pool, and agriculture in this small community and this blessing on the altar concept would come together to produce a pool of better and better crops over a very short period of time. When you're raising animals, it usually takes a few years to get your yield where you want it to be. Because animals have generation times, for instance, cows, and growth periods that require them to be mature before they can reproduce, that can take years sometimes just to get to the next generation. But when you're dealing with plants, they often happen to reproduce in a period of not just a year, but maybe even a few weeks in some cases. And so the amplification of certain traits can be done very rapidly. And with the guidance of a community or the oversight of a church and a religion to enforce it, always putting the best crops on the altar for an offering and a blessing, this sort of genetic modification became a thing that's driven by the community's rules. Now, recognize that this isn't science, even though it has a scientific result. It has a technical background behind it. Instead, it is magic, a belief in unknown, uncontrollable forces that we can't define, having an effect on how we live and the growth of our crops and the yields that we get. Nevertheless, the fact is they'd stumbled across a rule that worked well, and they went with it. They just didn't know why it worked. Now, while people understood that the parents of an organism had specific traits that could be carried on to the offspring, they didn't understand why, and no serious study of this was really made of any consequence until the middle 1800s by Gregor Mendel, who was a monk who decided to unravel this puzzle. Mendel is considered the father of genetics today, and he lived from 1822 to 1884, and he spent 8 years, from 1856 to 1863, growing about 10,000 different pea plants. And what he did was he selected them by their traits. For instance, yellow peas versus green peas, large pods versus small pods, wrinkly peas versus smooth peas. And by mixing them and looking at the traits that showed up in the offspring, and basically doing statistical analysis, he deduced some very simple laws, and one was the law of segregation. Each trait was defined by a pair of genes, and the parental genes were randomly separated. The sex cells contained only one gene of the pair, and the offspring inherited one gene from the parent and one gene from the other parent, and those two genes would end up in the plant. And then he also came up with something he called the Law of Independent Assortment. The genes for the traits are sorted separately so that the inheritance of one trait isn't dependent on the inheritance of another trait. In other words, your father may have dark hair and green eyes, and your mother have blonde hair and brown eyes, and it doesn't mean that you're going to get both traits from the parent. The traits are independent of each other. But Mendel also formulated another rule, and that's the rule of dominance. And this means that some genes are more likely to be expressed than other genes. So the rules of genetics themselves are actually pretty simple. You have sets of genes that come in pairs, and you get one half from one parent and one half from the other parent. And you have individual genes that are not connected so that different traits can emerge in any sort of a mix you might imagine. And you also have the fact that some genes will dominate, so that if you have a copy of a specific gene from a certain parent, it's more likely to be expressed than some other gene in some circumstances. The rules, again, are easy to understand, but it doesn't tell you what the underlying mechanism is. And it took a little while longer to figure that out, and that's when genetics came to be what it is. The real understanding came with the realization that the information had to be stored somewhere in the cell, and what a gene actually was. And it turns out that it is actually a string of molecules tied together, some chemistry that's done in such a way that it represents information. Now, what is the information? Well, basically, the information in your genes is essentially a series of instructions for producing proteins or enzymes or control genes. And those three things regulate and dominate. They control what materials are produced in your cells, and those materials act as structural materials to build the cell and its parts, or they act as the instructions via chemistry to have certain genes turned on or off and expressed or not expressed. And they also act as enzymes, active molecules that convert one sort of molecule into another. So in effect, there is a storage of data in every living cell known as genes. And the genes are organized into, think of them as file cabinets called chromosomes. And every cell, every living cell has this collection of file cabinets or chromosomes in its nucleus with all of the information and instructions for creating copies of itself. But it also has a lot of information in there that is stored away and is not expressed. In other words, any cell that you examine typically has a number of instructions that are expressed to make the cell what it is, and a greater number, usually around 90%, of instructions that are not used at all. And people were very puzzled about this for quite a while. Now, an obvious question is, where do all these genes come from? The ones that are expressed and the ones that aren't. Where did they all originate? The answer actually can be quite surprising. Many genes are created through mutation, and this happens for a number of reasons. Sometimes a mutation is created by an accident in copying the genes when the cell reproduces. Sometimes it's caused by a particle of radiation passing through and altering the gene. Sometimes it's caused by chemistry in the environment that forces mutations to happen, and there are many chemicals that will do this. Sometimes it occurs because of a viral infection, and the virus's insertion of genetic material in the cell changes what the cell is going to carry on to its offspring. So genes can actually be carried from a virus into a cell. But in all of this, when we're breeding plants or breeding animals, the genes that we use are the genes that are already present in those plants or animals. And when we mix different types of plants and animals, we'll discover something interesting. Some will crossbreed and some won't, and there are very simple reasons for that as well. But in the end, normal selective breeding happens between animals or between plants that have a known set of genes, and we're simply exploring those genes and how they're expressed. We're going to look at the mechanisms behind that, and then we're going to look at genetic modification and what it's all about and how it occurs. This is Talk Universe. In the science fiction movies, we often see some horrible accident where something gets crossed with something else and a monster emerges. And you wonder, what's the likelihood of a reptile sharing its genes with a fern? Why can't we cross a redwood with a fish? Why does it not work? Why is it that even similar-looking animals often can't crossbreed with other similar-looking animals? And the answers are really very simple. Our genes are arranged in very specific ways, and the rules of how they're arranged have a lot to do with it. In fact, everything to do with it. It's sort of similar to why a certain type of key will only work in a certain type of lock. There's literally a mechanical arrangement that controls it. Plants, on the average, have a certain number of chromosomes, and typically it's twice as many as what's present in an animal. And that might seem like something a little odd to understand. Why would a plant have more complex genetic information than an animal? The answer is really very simple. When times get tough, an animal can run away, can go somewhere else where times are easier. Plants have to tough it out wherever they're planted. If you rip a plant out by the roots, it's going to die. And so a plant, wherever it happens to be planted, wherever it's living, has to have enough tricks in its bag to handle whatever conditions nature throws at it. As a rule, plants have many more genes than animals, and this is why. Animals can flee, plants cannot. But what makes it impossible to cross a turtle and a frog, or a fish and a sparrow? It's because not only the number of chromosomes is what sets the tone, but also because of how the information is arranged in the chromosomes. So an animal with 12 chromosomes can't cross with an animal with 14 chromosomes or 20 chromosomes, because the number of genes that each parent would share doesn't match up, and it can't produce a viable offspring. That's the first rule to consider, the number of chromosomes. The second rule is you may have animals that look very similar and even have the same number of chromosomes, but they still can't crossbreed. And that's because where the instructions are for producing certain proteins or enzymes or control genes has to match in both parents. If the instructions are in different places, then the chromosomes, once again, can't fit together properly. The genes can't match and produce a viable offspring. Imagine taking the instructions for baking a cake and the instructions for building a power plant, and you know that the power plant will have many more pages and different materials. And imagine swapping a few of the pages and seeing what you get. You're not going to get anything you want. So what defines a species is whether or not it can interbreed with another element, another animal or plant. And if that breeding is rare or impossible, then it's a different species, or even a different subspecies. Sometimes with persistence, you can get animals that are similar to interbreed, but often the offspring will be sterile or different. Think about the difference between a horse and a donkey, and how when they breed, they produce a sterile offspring known as a mule. This is because while they're close enough to sometimes produce, they won't produce an offspring that has the capability of producing offspring of its own. So genetics has a certain set of rules, and they're typically hard and fast, but sometimes an exception can be found. You're not going to find a giant lizard crossing with a daisy or something along these lines, specifically because the number of chromosomes or the arrangements of the genes, everything is different in those organisms. And the proteins that are produced are different and will have a message of their own. Chemistry is the message of a cell. Cells communicate with each other with chemistry or with proteins. And proteins are chemicals as well, but very complex ones. When we have a drug that's introduced into a system and it changes how that system works, it's because the drug is acting as a message or a regulator to the cells, telling it what to do. The cells respond in kind. Each chemical is a switch or a message that tells what actions the cell should take, or what actions the system should take. When you consider something like pituitary hormone, which causes you to grow taller, then you know that that hormone is carrying a message to the cells and the tissues, telling them how to respond. When you get drugs in your water and the drugs affect how organisms live, it's because those drugs are messages to the cells in the organism. So it isn't difficult to understand. However, even though we can know why an animal can't be crossbred with a plant, or even similar organisms often can't interbreed, there are other rules as well. In cells in nature, typically genes don't move from species to species, and yet it was discovered that with certain types of viruses, this could happen. A virus actually injects genetic material into a cell, and if it happens to be a cell that is a germ cell, such as an egg or a sperm, then what will happen is radically different than if it were to infect something like the tissue in your arm. You might grow a wart or a tumor, but if it is an egg cell or a sperm cell, whatever information was injected will be carried to every daughter cell that is grown in that tissue, and if this cell is the beginning of a new life, the beginning of a baby, then what will happen is every cell in that child will have the information injected by that virus. This is how new organisms are made, and how mutations are carried from generation to generation. All the cells in your body can mutate, but only if it happens to be in an egg cell or a sperm cell will that mutation carry to the next generation. Think about how unlikely that is. In genetic engineering, we often find that we can inject genes from one thing into another and see them expressed. This is normally done in plants because plants can be grown from a bud of tissue known as callus. Callus tissue is not differentiated. It isn't stem, it isn't fruit, it isn't leaf, it isn't the root of the plant. But it's something that's generic, sort of like the scraping of the cells inside your mouth, epithelial cells. Callus tissue can form almost any structure, and it's very easy to work with. So whenever we're making a new plant, we often take a scraping of cells from a plant, we break the cell walls, we introduce new genes, and then they reform cell walls, and these new cells grow callus tissue, which is often grown in a petri dish. Callus tissue is then divided up with a scalpel, and each bud of the callus is placed in its own growth medium, and it's treated with something to make it differentiate. If you've ever used Rootone, the material used to root plants in your garden, you see a compound, naphthalene acetic acid is what it's called, or some derivative of that, which acts to tell the cells, as a message, to begin growing roots. If you take callus tissue and treat it with Rootone or a similar compound, what will happen is it will begin to grow roots, and this message to the rest of the tissue tells it to begin growing stems and leaves. This is how the new plants are formed, and they're very similar to something grown from a seed. But these plants will have the genes that you've injected in them and can easily propagate to hundreds or dozens of copies, whatever you need for the experiment you're doing, and can often show new traits in these plants from the manipulation that you've done. What's very different about this from crossbreeding things is this. When you interbreed different plants or different animals, you're using only the genes that are present. When you do genetic manipulation or modification, you're literally writing a new gene or injecting a gene from something else. And this is done in a number of ways. One method is to literally inject those genetic messages into a virus and infect some cells. That's a little difficult to do sometimes. Another method is much more direct. You create a new gene, or you have copies of a gene, and you put those copies on microscopic tungsten beads or pellets, and then they're injected at high speed under pressure from something like a gas gun, and they perforate the cell. And these tiny pellets carry the gene into the cells, just like bullets. When the cell heals, it incorporates the genes into its own genetic structure. And if it is successful, those cells will now have your edited genetic message inside them. Now, as odd as it sounds, this high-tech method was actually related to something that happens with certain species of biting flies. These flies can literally tear apart the cells in the host animals that they bite and carry some of their material to the next subject that they bite. And in this process, sometimes genes are carried from one species to another through the fly's bite. Because the cells that they bite split open, some of the genetic material is stuck on the fly's mandibles. And when the fly bites the next subject, some of that material is carried to other new host cells. So in the wound, some of these tissues show transgenic effects, the effects of carrying genes from one species to another. This is exactly the effect that's being duplicated with the tiny tungsten beads. Now, as I mentioned earlier, you can't simply crossbreed one creature with another without following the rules. After all, if this were true, then chimeras, the interbreeding of different types or species, would be very common in nature. We don't see them because of the fact that reproduction requires that the sets of genes match properly, the right number of chromosomes and the right structures of chromosomes. But when you take individual genes or short segments of genetic material and introduce it into a new cell, something odd happens. That strand of material generally forms a loop called an intron. This is a short sequence of genes
that have been moved into the cell and sometimes is used to communicate genes from one chromosome to another. Now, in nature, cells typically have a specific set of chromosomes, a number of chromosomes, for each type of species or each type of organism that there is. But this isn't always a hard and fast rule. Sometimes errors can occur when cells divide. In some cases, you'll have a simple cell, and during the division, something will go wrong, and the cell will end up with extra chromosomes. This might sound like something that would paralyze the development of an organism or stop it altogether. But in some cases, an extra copy of a chromosome doesn't terribly interfere with the action or the ability of the organism to survive. In this case, something odd will happen. Now the cell has more file cabinets to store information, and over time mutations may accumulate in that and in the original chromosome as well. Sometimes what will happen is the organism will show greater complexity because it has more information storage and more genes that can be expressed. Some of the genes will be turned on or off at random over the course of time, and some may affect how the organism lives and develops. Sometimes this leads to the differentiation and development of actually new species. Because this new organism can't reproduce with other organisms in a sexual manner, because its number of chromosomes is wrong, you may think that's the end of the line. For simple things like mushrooms or blue-green algae, this isn't such a big issue, but for more complex organisms, it can be the end of a new generated species. But in nature, there is a process where organisms can sometimes reproduce without the need for sex. In higher organisms, this is called parthenogenesis. This generally only happens in the female of the species because they're the only one that can actually raise their young internally or lay eggs. So sometimes it's possible for an organism to have a radical mutation, a change in number of chromosomes, to appear very much like the original organism, and yet to successfully reproduce. This is often seen in fish, frogs, and lizards. Parthenogenesis commonly happens in times when their environment shows them great stress and they're unable to find a mate. So some organisms, some higher animals, can actually reproduce without a mate under these circumstances. Now, with all this being said, we see that if we were to edit genes directly or to carry genes from one species to another, just how well it's going to reproduce on its own is often an issue. But when we start working with crops, things that generally reproduce on a short time scale, such as a number of weeks or months, we often find that by using the explant method I mentioned earlier, growing crops from a bit of callus tissue, it's possible to make hundreds or thousands of copies, and they become established, in effect, as a subspecies or their own type of life form. Usually, however, what will happen is an organism will be engineered or altered genetically, not by crossbreeding, but by introducing the genes in the methods that I mentioned before. And in this case, usually one or two specific genes are placed in the organism that can easily interbreed with the standard organism as well, thus spreading the genetic modification to the organism that they started from. This becomes a hybrid of genetic modification and then selective breeding to amplify or propagate the gene that was added. The main danger from this is the irresponsibility of allowing untested or unproven genes to escape into the wild and contaminate the original stock. Now you no longer have the original material that you started with. The other issue is, as we add more genes, we don't know what sort of interaction can occur when those genes are activated together. It could actually create a situation where unwanted effects occur and spread throughout the wild population as well. In a very short order, some forms of our food crops could be irreversibly contaminated, and nobody would know what to do about it. We could lose foods that we have cultivated from their original stocks over the thousands of years to a simple contamination act and be unable to recover our original stance. We literally could lose some of our basic food stocks, or they could become poisonous, or unable to be eaten, or actually could become noxious weeds. But what happens when you're growing a natural crop and pollen from a genetically modified crop ends up on your field? In some cases, farmers who had no idea that this was happening have been sued for growing crops that contain genes that they didn't want and they didn't ask for. So how do you protect yourself from being sued for something you didn't want and you didn't know was happening? We'll talk about this after the break. This is Talk Universe. So how did we figure out how to do genetic modification of any organism? A lot of it started a few years back, after an understanding of what the genetic code was about and how the genetic molecules were configured. A fellow from India, a researcher with the name of Chakrabarty, decided to see if he could modify simple bacteria to destroy the defoliant 2,4-D. Now, this is an extremely powerful defoliant, one capable of killing many organisms very easily. And so the question was, could an organism actually adapt to that and live with it? He began by using simple methods, selective breeding. He used a dilute solution of the defoliant, which killed almost all of his subject organisms, but a handful did survive. He selected them and bred them, and when they were healthy, gave them another dose of the defoliant. Over time, they became quite tolerant to it, but they reached a level where they didn't seem to be able to go any further. He needed to accelerate his changes. He used various mutagen compounds and other methods to cause mutations to occur within those bacteria. After a while of selective breeding, mutation, and more selective breeding, he arrived at an organism that was capable of living completely on 2,4-D, actually synthesizing everything it needed from the defoliant and living successfully on it. The organism could also live very successfully in the wild. So he had an organism that he had created by forcing evolution to occur in the laboratory, and created something that was able to actually clean up the defoliant in areas where it wasn't wanted. This is a great advancement, and an understanding of how breeding and genetic modification could occur. So by using genetic modification in conjunction with selective breeding, he was able to reach his goal. What's important to realize, however, is that mutations of this sort are done completely at random. Using millions of organisms and hitting them with tough conditions and seeing which ones survive is not in any way a control of genetics. It is only the results of natural mutations or induced mutations and selecting the most fit organisms for your experiments. It wasn't until the genetic code was fully understood and what it spelled out that we knew that it was making proteins, enzymes, and control genes. And only at this time could we begin to write our own genes from scratch. But writing a gene isn't as simple as putting the symbols together to make the cell produce a protein. You somehow have to get that gene into the cell, and that's where the tungsten beads or viral infection methods step in. This still leaves us with the question of why we would genetically modify an organism. There can be a number of reasons. For instance, we might want to make a food crop grow in an arid region, or in the case of, let's say, short-stem wheat, a lot of the energy of the wheat plant's growth is in the length of the stem. But if you could make it grow short stems, then all that energy could grow into more wheat instead. And that's exactly what has been done in some cases through selective breeding. But with genetic modification, you can make a deliberate change now in a very short period of time and have your results in a matter of weeks in some cases. And other reasons are beyond speed or time of reproduction. For instance, no natural rose has a gene for blue coloration, and yet we can take the blue coloration gene from another plant that's compatible, inject it in roses, and grow blue roses. From a practical standpoint, this isn't really anything, but when you get down to something like a crop that grows readily and yet isn't fully nutritious, you see the possibility for adding the gene for producing lysine, an amino acid necessary for human nutrition, grow in that crop, could be beneficial. This is what's been done with corn. Corn doesn't produce proteins that contain lysine, which is essential to human nutrition. And we can inject the genes to grow lysine-rich proteins in the corn and grow it with no other apparent effect. This creates a crop that is actually more nutritious and grows just as readily as corn does anywhere in the world. So here's a clear case where a genetically modified organism could actually be of benefit. Similar methods could produce bacteria that make spider silk, which could be used to produce it in bulk and make fabrics that are extremely light, tough, and even bulletproof. Other modifications could include bacteria or simple plants making plastic for us instead of consuming petroleum to do the job. But in any beneficial technology, we also run into the dark side of things where people do modifications that were not originally thought to be even ethical. One choice was made to include certain human genes in a variety of rice, which is now being sold all over the world. And it's raising questions of cannibalism. If you're eating human proteins in a food that were introduced there by genetic manipulation, are you in fact eating something that is human? A similar case arose in producing gelatin that uses human proteins. It was claimed that because some people have a reaction to eating gelatin made from cow or pig products, that if we used human products instead, it wouldn't create this sort of reaction. But now you are eating a human protein. This raises serious ethical questions that must be addressed. And it also begs another question. Who's monitoring what's going on and who decides what's ethical for you? The issue, simply put, is that we're breaking down a barrier. There used to be a very distinct line where you do not eat human beings. But now, by introducing a protein here and an enzyme there and a chemical here or there, we're slowly eroding the boundary between eating foods and being cannibals. And this is something that has to be discussed and stopped. The use of genetic engineering in our foods actually began before we were modifying food crops per se. And this has happened because a number of organisms contribute to the success or failure of growing a crop. One is a type of bacterium known as Pseudomonas syringii, which is growing on the roots of certain potatoes and lower on the stems. What happens with this particular bacterium is the shape of its proteins on its surface encourages the formation of ice crystals and frost when the weather is cold. By changing the structure of the outer proteins of this bacterium and then introducing it into the fields, the new bacterium did not encourage the formation of ice crystals, and it actually limited the number of plants that would freeze when the weather got cold. But one of the unexpected consequences of this is the bacterium is often swept into the upper atmosphere. Cloud formation occurs because of nucleation. This is the formation of ice or water droplets on tiny particles such as dust or bacteria. If the naturally occurring bacteria dominate, then it's much more likely that ice crystals will form at certain altitudes, leading to the formation of more clouds. And yet this modified bacterium doesn't have that ability because it doesn't encourage the formation of those ice crystals. Does this, in fact, constitute geoengineering? Does it change our weather because this modified bacterium meant to keep the potatoes from freezing is actually limiting the formation of clouds? With this and other examples, our world has now become a home to hundreds of modified organisms that are having unexpected effects on our environment. And some of them are very subtle and may take many years to show up. How do we reverse something when we don't even know what's causing it or what the effect might be? Normally, these sort of concerns would be restricted to research labs or large corporations, but recently there have been some interesting twists that have occurred, and one of them is Crispr Technology. This is a method of inserting small genetic sequences in a cell and doing the editing under very prosaic conditions. You have the ability to find repeating sections of RNA and insert spacers with your genetic message into these sections. This information is then taken in by the cell and editing becomes possible. People can edit most any organism using CRISPR methods. What this brings us to is a time when most anybody with a little bit of genetic wherewithal can actually produce tailored organisms at home. This is not limited to simple things. Some people have already edited mushrooms which will be sold as food. You can only imagine the sort of places that this could take us. The ability to create organisms that make custom materials on demand is actually within our grasp today, and it can happen in a very simple surrounding. But what drove large companies to manufacture genetically modified organisms, and where do we stand with it today? The predominant type of organism is one that has been engineered to withstand the use of weed killers. Everybody is familiar with Roundup, a material that is a defoliant that can destroy most types of plant life easily. When performing agriculture, it's necessary to do a lot of weeding to control what happens to the growth of your plants. If you don't weed your fields, the weeds will dominate and take over very rapidly. Your yields of crop will plummet, and you won't make any money, and you certainly won't produce much food. So the thought was, if you had a food crop that had the ability to resist a weed killer and could grow even when it was used in its area, you'd have a winning combination. But unintended consequences soon followed. For one thing, many organisms developed a resistance to the defoliant very rapidly, which meant that it wasn't quite as effective. For another, the plants in question began to be dosed with this defoliant, and even after a period of time when the defoliant levels were supposed to be safe, and after washing, some of it was still retained on many of the vegetables that made it to market. People were being exposed to levels of this defoliant, and new ailments were showing up, and at this point the controversy is whether the defoliant or some other effect is causing them. One is the sudden surge of gluten intolerance. Many people suddenly around the world are showing an intolerance to gluten. Wheat, which is a major source of gluten, has been in our diet for approximately 50,000 years. During that period of time, nobody or very, very few people have shown an intolerance. However, at this point, it is somehow believed that all over the world everyone has suddenly become intolerant to this protein which we all consume. No mechanism would make everyone on Earth suddenly intolerant. It is far more reasonable that something else in our wheat supply is causing this reaction. And in fact, it appears that some of the fragments or products of the breakdown of this defoliant are responsible. Other researchers are gathering evidence that this defoliant, when it gets in our system, interferes with the activity of normal gut bacteria, thus inhibiting the production of compounds that we normally have in our metabolisms. The question isn't fully resolved at this point, but it's pretty certain that you wouldn't want to take a compound used to kill most biological material, such as plant growth, and put it in your mouth. But another issue of responsibility emerges here. If you were going to modify plants, and you were going to do it in such a way that you were putting these on the market, wouldn't it make the most sense to start by modifying a version of the plant so that it could not interbreed with the original stock, thereby assuring that the germline remains clean? In other words, wouldn't you edit a plant that can't possibly put those edits back into nature? That way you could retain control over its spread and exactly what happens to the genes that you have created. This isn't a difficult thing to do. You see, there's a certain structure to genes, and those structures have to match properly in order for a plant to breed with others of its type. If you alter the order of the genes or the structure of the chromosome slightly, then those plants are unable to breed with each other. It would not be a difficult task to reorder some of the structures of the chromosomes so that these plants can't possibly breed with the other plants that exist in our food supply, and we would retain full control over how this plant grows and what it does. But no biotechnology company to date has done anything even remotely like this. It only makes sense when you're working on the files on your computer that you make backup copies in case you make irreversible changes. Why then have we not pursued the technology that allows us to have clean copies of our natural heritage of foods and take care of this possibility so that we don't wipe out the originals? What may surprise a lot of people is how closely many of our crops are related to each other. For instance, anybody who knows what kohlrabi is, or kale, or broccoli, or Brussels sprouts, or cabbage, or cauliflower, may be unaware of the fact that they are all varieties descended from one single plant, wild mustard. Wild mustard has some specific value as a food crop, but it also has small structures and doesn't grow very much. Kohlrabi is a modified form of wild mustard that has a much larger stem. It looks very much like a root vegetable. Kale, of course, is a version that is modified for the growth of its leaves. Broccoli is a form of wild mustard that has been bred to have large numbers of florets and thick stems that are edible. On Brussels sprouts, the lateral leaf buds have been bred to be very large. Cabbage, of course, is the terminal leaf buds, the head itself. Cauliflower is a variety similar to cabbage and is specialized for its large flowery tops. The same can be said for eggplant and tomatoes and many other varieties of crop. We'll find, if we look into the history of corn, that it actually is descended from a grass-like plant known as teosinte. And only after many, many generations of crossbreeding were they able to bring it to the state that it has today. So many of our food crops come from only a handful of small initial crops, and depending on how they were grown and bred in local areas, arrived at certain varieties that we accept today. Later, as commerce and world travel grew, new varieties were created by crossbreeding the existing varieties. So without the use of genetic modification at all, and only the use of natural breeding methods, we were able to create almost every one of the vegetables and plants that we have in our larders today. Now, this is not to say that we don't need genetic modification, because we certainly can make plants that do things that have never been done before. And increasing the nutritional value and adding amino acids that we can't get normally certainly will be a help in world population problems today. If we really wanted to have a positive effect on our food production, we'd focus on plants that produce more nutritious food and do it more compactly, with less resources. But one of the facts about farming is this. Generally, when a plant is very high yield, it tends to have very specific requirements that can be difficult to meet in nature without a great deal of effort. We need cleaner water, better fertilizers, and better soil as a rule. This isn't always the case, but we should focus on plants that yield more with less effort. We'll be back after the break. When we start modifying the structure of the world around us, we can quickly run into the fact that we really don't know a whole lot about it. Everything we know has been learned by experimentation, by trial and error, and we often see disaster as a result. We have to be exceptionally careful when we start tampering with the basic genetic instructions that make up all of the things around us in our world. It's very easy to insert instructions that have unintended consequences. This happens in systems as simple as computers. The thing on your wrist, the thing in your pocket, the thing on the desk. We often insert commands or give it instructions that it follows to the letter and does something extremely different from what we intended. How much more so in something as complicated as an organism? When we start tampering with our food supply, and we start tampering with the microbes around us in our world, we have to be ready for things to happen that we never intended or never wanted. There are sure to be some benefits, but the fact is, when you tamper with a complicated system, you're more liable to break it or cause something to go wrong. In our world today, it makes far more sense to concentrate on making robotic or laser-based defenses for insect attacks on plants. For instance, there are already machines that recognize the wing flap patterns of a mosquito and destroy them with laser light. This is something that does not use pesticides, doesn't damage the environment, and is fairly small and easy to produce. Similar systems could be made for keeping insects, such as moths or caterpillars, off of plants. There are already talks about making robotic bees, insects to pollinate, because of declining bee populations, colony collapse. And yet nobody seems to look at the fact that similar robotic guardians placed on crops could do exactly the same job with the use of no pesticides whatsoever. This would alleviate most of the needs for pesticides in many cases, and is a very sound and safe alternative. And I haven't even talked on the genetic modification of animals yet. You've probably seen some of the pictures on some of the less reputable sites of chickens with no feathers or no beaks, used to produce chicken nuggets, things along those lines. Now, a lot of that is very questionable material, but there is some truth to the fact that they are modifying animals to meet the needs and not spend their energy growing parts that aren't going to end up in your food. Is it moral to create an animal that is meant to be a food beast? Is it moral to create something that actually has a brain and eyes that's going to be nothing more than something we grow for the table and has no chance whatsoever of surviving in the wild? When we reach the ability where we can do things of this nature, we have to question our own moral basis. I'm not a vegan, and I'm not a vegetarian, and I enjoy a good steak like a lot of people. I eat meat. But I must say that if we have the opportunity to produce foods that are the same as meat without killing any animals, then it makes the most sense, and this would be the place where we should aim our genetic prowess. We should learn how to make things that are morally responsible. If it means making a vegetable substitute for meat, or it can be actually meat growing in a plant, those things can be done. It's difficult, but it's something that we should look at because it would eliminate a lot of suffering for the world. When you consider the enormous resources it takes to produce meat, and the amount of animals, the sheer numbers of animals used to do it today, then it stands out very clearly that the best use of our resources would be to find some method of doing it far more efficiently. Now, meat is an energy-intensive product. It takes a lot of specialized materials and a lot of growth to do. The volume of water used alone is staggering, and the amount of land used to raise the food for the meat is staggering. But you see, plants are basic energy gatherers at the very bottom of the food chain. They are actually somewhere above the microbes, of course. The microbes in our soil and in the oceans, the microorganisms, are the ones that do the basic gathering of materials and energy. But if we could genetically engineer a plant to be more efficient, because most have a photosynthetic efficiency of only 1 to 1.5%. If we could double that, we could double food production and double the capturing of carbon dioxide out of the atmosphere. Here are two goals that genetic engineering should be aimed at: plants that are far more efficient in their growth and capturing and fixing carbon. If we could come up with something as simple as that, we could change the food equation on this planet easily, and we could use bioremediation, organisms designed specifically to clean up the environment to help save us from a great deal of grief in the world today. But here we arrive at a potential danger. Very few of us would trust a politician or a big company to do anything properly. And one of the issues that we really must face is we don't know what's in a lot of our food today. I don't want a company that makes drugs and pesticides producing my food. It makes the most sense for someone, a disinterested third party, to oversee whether there is a conflict of interest in the companies that are producing poisons and food and how they go about marketing those things to us. If we produce successful meat substitutes from biology through genetic manipulation, it sounds like it would be a godsend. But the issue is this: I want to know what's in my food. I want to know how it's produced. And I don't necessarily trust that a big company that makes synthetic meat won't put something in it that I don't want. The issues are very simple. Most people aren't very well educated about these things. Younger people are becoming more so because they have a great access to information that we didn't have in our time. And yet, we do know enough to know that there are certain things we certainly shouldn't have in our foods, and we don't want them there. The cannibalism issue is only the tip of the iceberg. Including human genes in our food should never be done. After all, we don't have to eat people to stay healthy and alive. If somebody does this specifically because it makes it easier to produce or makes them a buck, they're certainly on the wrong side of the fence. Now, my book recommendation is an old one, but one that I read at age 8 and I thought was astounding. The book is Brave New World by Aldous Huxley. It was written in 1931. And it's set in the future where genetics has been used to create specific sections of the population, and people are essentially controlled by medications. It's an amazing, amazing book that predates many of the things we see in the world now. Everything is monitored and controlled. And it's amazing in that he foresaw many of the issues that we are facing today very clearly. Our Singularity Watch is aimed at the fact that there are a number of devices in our lives that we depend on, including health devices, and all of them are too hackable. It's been discovered that many of the devices that are implanted in people, including medical pumps and other devices, can be hacked into and reprogrammed by malicious people. Unfortunately, there's very little that can be done at this time. What really needs to happen is encryption standards and some sort of security system need to be in place on every one of these devices. There's a fellow by the name of Mark Goodman, who is the Singularity University's faculty chair for policy, law, and ethics. He gave a keynote speak at the Exponential Medicine Conference, and he talked about medical security and privacy threats in healthcare and what we can do about them. It's interesting because these sorts of devices have been around for many years, and with the explosive growth of computing, nobody seems to have looked at the security measures that should be in place in medical devices. Goodman points out the connection between big data and analytics and how popular it is with criminals as well as law enforcement. And hackers go after medical records because it's worth up to 10 times more than a credit card number on the black market. There's so much information in them that is actually worth money to a criminal. It's now said that we're going to see amazing changes, 8 methods that AI will profoundly change city life by 2030. There was a report just created, the 100-year study on artificial intelligence by Eric Horvitz, a computer scientist, and the former president of the Association for the Advancement of Artificial Intelligence, and he's also the managing director of Microsoft Research's main Redmond laboratory. In this, he studies the effects that AI will have on our society, and at the top of the list is transportation. We're now seeing artificially intelligent-driven vehicles everywhere. They'll be widely adapted by 2020 in many cases, he feels, and driverless delivery trucks and autonomous delivery drones are going to be at the top of the list. Home and service robots are also expected to make an appearance very shortly. By 2025, it's expected that there will be many cloud-connected robots serving our homes and taking care of our needs for us. In health, where AI systems are now becoming more trusted for diagnosis than many doctors, and this is expected to continue. Education is another field. A lot of people have difficulty relating to some teachers, and if they had an AI that worked with them like a friend and understood their needs, it would be far more effective in teaching them in many cases. We expect to see huge increases in the use of AI in education very shortly. To see the entire article, go online to the singularityhub.com website. They have a number of Singularity news items there that are extremely interesting. I'm going to wrap this up with a few notes. We really need to think about the future we're creating, and genetic modification is one of those tools that can produce amazing benefits or amazing horrors. We need to be very cautious about where we stand morally when we start modifying organisms and what it can mean to the community around us. Our world sometimes has what appears to be a tenuous hold on life. This isn't true. Life will persist no matter what we do. What is really at stake is human existence, not life in general. We may wreck the world. We may do things that decimate life. But what will die will be humanity and the methods of supporting it. Life will continue, and at some point the world will emerge with other large animals, other forms of life that we can't imagine. But we will be gone. We need to look at what our goals are. What are we trying to create in the world? If our aim is to destroy things, we seem to be well on that course. If our aim is to improve life and make it better, we have to decide what that means. If we're serving the needs of the people, then we need to make products that are usable by the people and do things that people need. But one of our fallacies is the picture that we must have some sort of an economy as we see today. The reality of the matter is, all we really need is food, clothing, and shelter, and some sort of social connection in order to survive. Most people aren't interested in being great researchers or inventors or scientists. They're interested in living their lives and living well, being comfortable. Most of the things that we have in our technology are intended initially to make life easier and better, but oftentimes become much worse. How many times have you bought something that was supposed to be a new, improved version of something, but it was not any better, it was just different? They substituted the metal clips with plastic pieces that would break. They made parts that would wear out faster. You end up with something that costs a little less, but ends up in the landfill and doesn't do the job you need. We've got to focus on how we design things, what their purpose is supposed to be, and how we are going to recycle them in the end. After all of the things that we are making end up somewhere in the world in some form or another when it's all used up and done. Genetically modifying things gives us so many more abilities. We could improve the photosynthetic efficiency of plants. We could come up with ways of creating foods that are more nutritious, things that grow in horrible environments very easily, and systems that can actually clean up the pollution we've created in the world. We have the opportunities and we have the tools in our hands, but we have to figure out what our priorities are and where we're going to focus. Companies are often focused at the bottom line and are unwilling to participate in projects that don't yield an immediate benefit for their stockholders. Governments often lose track or focus and seem to become corrupted very easily. Pretty soon they're drawing in so much money and doing things with it in our name that we never authorized. Putting this sort of project in somebody's hands sounds like a very difficult task at this point. We don't seem to have a third disinterested party capable of carrying out these projects without becoming corrupted or losing sight of their focus. And charitable organizations seem to be one of the worst possible models for this. Look at the scandals involved and the huge amounts of money brought in for relief funds and how little of it actually reaches the people who need it. So what we need first and foremost is to find some way of getting our goals outlined clearly and finding some method, some organization or entity that can bring it to a focus and execute it properly in the interest of the public and in the interest of humanity. This is probably our most difficult task. After all, we already know how to solve most of the problems that we have, and much of it is based on human greed and evil. Self-interest can be a really powerful force in somebody's life, but when it is used to the point where it is to the detriment of others, something has to be done about it. Now, the establishment of genetic seed banks and gene banks is an essential part of this equation when we start beginning to modify organisms. When these things escape into the environment, and already many have, we know that there will be consequences we're unaware of for some period of time. We don't know how to fix this. We don't know if it can be fixed. But we do know that we have growing technology and we don't have growing morality to match it. We need to be responsible, and we need to take responsibility for the decisions we make. Otherwise, we will end up destroying the world. Faceless organizations such as governments, industries, and committees absolutely cannot be the forces that do this. We've got to solve the problem of accountability and how to execute a program like this successfully and for the betterment of humanity. Remember my one thought that I always share. We have everything we need to create everything we will ever need. I believe that more now than ever, and I see the growth of our information, our knowledge, and our experience. I hope that all of us will do the best we can with our lives and help everyone. I'm Sir Charles Schultz. This has been Talk Universe. Thank you for listening.